Good morning, everyone, and welcome to the fifth webinar in the series that Department of Biotechnology is organizing in association with DBT Wellcome Trust India Alliance to highlight the role played by DBT, its autonomous institutes, and the public sector undertakings in mitigating the COVID-19 pandemic. My name is Dinakar. I work with DBT Wellcome Trust India Alliance, and I'll be your host for today's webinar. While the first webinar in the series had Dr. Renu Swarup, Secretary DBT, Dr. Alka Sharma, Advisor DBT, and Dr. Kavita Singh, Mission Director, National Biopharma Mission by RAC, as experts to discuss DBT's multi-pronged research strategy and action plan to address COVID-19. The second webinar focused mostly on how DBT is leveraging the startup and industry ecosystem to respond to the pandemic, and how startups can be a part of the efforts. We had Dr. Manish Diwan, Head of SPED by RAC, Dr. Taslim Arif Sayed, CEO CCAM, and Dr. Jitendra Sharma, CEO Andhra Pradesh MedTech Zone as the experts. After the first two webinars, we have also conducted another two webinars talking about the role of the Autonomous Institutes. The third webinar with Dr. Soumitra Das, Director, National Institute of Biomedical Genomics, Dr. K. Thangaraj, Director of Center for DNA Fingerprinting and Diagnostics, and Dr. M.K. Bhatt, Director, National Center for Cell Sciences, as panelists, highlighted the efforts of the respective institutes in the development of diagnostics, vaccines, and drugs. In the fourth webinar, we met Dr. Amulya Panda, Director of National Institute of Immunology, Dr. Ajay Kumar Pareda, Director of Institute of Life Sciences, and Professor Pulak Mukherjee, Director of Institute of Bioresources and Sustainable Development, to discuss the role of their respective institutes in the country's response to COVID-19. The Autonomous Institutes of DBT have been at the forefront in designing science and biotech solutions to tackle this health challenge, this global health challenge of COVID-19. Their efforts range from development of diagnostics, vaccines, drugs, to training the medical professionals. Through these webinars, we have been trying our best to learn more about the efforts of the Autonomous Institutes of DBT. And today, in the fifth webinar of this series, we have with us Professor Sudhanshu Vrati, Executive Director, Regional Center of Biotechnology, Faridabad, Dr. Subeer Majumdar, Director, Translational Health Science and Technology Institute, Faridabad, and National Institute of Animal Biotechnology, Hyderabad, Professor Dinakar Salunke, Director of International Center of Genetic Engineering and Biotechnology, Delhi. I welcome you all to this webinar today, and thank you for joining us today. So before we begin with the presentations, I would like to just make a few housekeeping announcements so that we can take this webinar forward smoothly. This webinar will be recorded. This is for everybody's information, and the recording of the webinar will be available on the YouTube channel of India Alliance. We would request you to share your questions for our speakers today in the question box. You will find the question box in your console. These questions will be taken up by our speakers at the end of all the three presentations. In case the webinar ends abruptly due to any sort of technical glitch, you should be able to join back the webinar using the original link that you used to join this webinar. Lastly, if you want to share about the webinar on social media, do consider tagging DBT and India Alliance and the speakers, and use the hashtag DBT Webinars COVID-19. And with those few housekeeping announcements, I would like to invite our first speaker for today's webinar, Professor Vrati, to talk about the action at RCB in response to COVID-19. Over to you, Professor Vrati.
I begin by thanking the organizers for having me here. Today, I shall be talking about the development of antivirals against SARS coronavirus 2 and what has been the role of DBT-aided autonomous institutions in India. So we know a great deal about vaccines, and we know that vaccines are our best bet against infectious diseases. Vaccines work well when we know the pathogen, and these will protect us against the known pathogens. However, when a new pathogen emerges, we don't have a ready-made vaccine. Then making vaccine is a long-drawn process which takes several years. And even if you then have a vaccine, it would not work immediately because it takes a while for immune responses to kick in. This is precisely the situation that we are in today. We have a new pathogen which is going berserk, literally. That is the SARS coronavirus 2. We don't have a vaccine. So what is needed? You really need something that can work immediately to affect the virus that causes the disease. And these are the antivirals that can be potentially used to inhibit virus replication. We therefore need these antivirals, which could be used as a prophylactics or as therapeutics. And they should work almost immediately. Of course, they need to be cost effective. There are three kinds of drugs that will be useful to treat COVID-19 illness. Number one, those drugs which block virus entry into the cell, the drugs that prevent virus replication inside the cell, and the drugs that would minimize the damage that the virus causes to the organ systems in the human body. A number of strategies are being pursued across the world, including in several labs in India, to develop potential antivirals and drugs against coronavirus 2. These are listed over here, the foremost being the repurposing of existing drugs. But before I tell you more about this business of repurposing the drug, let me tell you a little bit about the drug development pipeline. It's a long pipeline. You start with developing a drug molecule or potential drug molecule, either in silico or in your lab through screening of library or designing a chemical inhibitor. You test it in the cell culture model, then in the animal model. And if it works, you get into the preclinical development and clinical development. And finally, in the clinical development, if it's proved effective, you have a drug. But if it doesn't, then you start all over again and start the pipeline. This long pipeline takes very, very long time, anywhere around 10 years. It's very expensive. The development of a typical drug cost about 2.5 billion US dollar. And chance of success are very, very low, which is about 5% or actually less than 5% of all the serious attempts. So a new strategy has been in place for some years now. We have about 1,500 drug molecules that are approved for human use against different indications. So when we say repurposing, we test the existing drug molecules for the new indication. In this case, for example, these drug molecules that are already approved for human use are being tested to inhibit coronavirus 2 replication and for use in the COVID patients. A number of such drug molecules are at various stages of evaluation or use, and I will talk to you about each of these molecules in some details. The other strategies are in vitro screening for virus inhibitors. There are several small molecule libraries that are commercially available, which contain several thousand compounds, which are drug-like molecules. You could screen those molecules in a cell culture model to test whether they could inhibit virus replication. A recent approach to drug development has been use of high-power computing, where you design molecules in silico or in computer, or you identify existing molecules that could be used to block the function of one or more viral proteins that are critical for virus replication. In last 6 months or so, a large number of such molecules have been identified in different laboratories, including in several Indian laboratories, including a few such molecules at RCB. We identified almost about 4 months ago methylcobalamin, that is vitamin B12, as a potential molecule that could inhibit the RNA-dependent RNA polymerase of SARS coronavirus 2. But when we tested this in the in vitro cell culture model, the inhibition was about 50 to 60%, which was way low compared to remdesivir, which we use as a positive control, which gives 99.99% inhibition. So a large molecules that were identified as being effective in silico have turned out to be not so in real testing in cell culture model or the animal models. Then we have therapeutic antibodies, and that is based on the premise that most viruses can be effectively neutralized for their biological activity by neutralizing antibodies. So if we have highly specific, high affinity neutralizing antibodies to a certain pathogen, or in this particular case virus, we could use them for treatment purpose. In India and China specifically, a large number of herbal molecules or preparations are also being tested as potential antivirals. Remdesivir is the molecule of choice or the drug of choice currently. This is a nucleoside analog. Originally, it was developed to treat hepatitis C. This was then used for treating Ebola, and it was tested in rhesus monkeys. But finally, when the clinical trials took place, it was found to be not very useful either for hepatitis C or Ebola. However, this drug showed antiviral activities against SARS coronavirus and MERS coronavirus. And based on this information, this drug was used in patients earlier during the outbreak in China. And there were mixed results that were coming out from China and other places which were using remdesivir. Sometimes they were saying that it is effective. In some cases, they did not find effect to be statistically significant. Finally, WHO took up this drug and carried out its evaluation under its solidarity trial. That trial was conducted at NIH, and the trial suggested that remdesivir reduced the recovery time of serious COVID patients from 15 days to 11 days, and also gave some survival benefit. Based on this, this drug was being authorized for emergency use in US and in India, and for compassionate use in Europe. Several companies in India have launched generic version of the drug, and these are, you can see in the picture, Zydus Cadila, Cipla, Jubilant, Mylan, Heterolabs. They have all produced various generic forms of remdesivir, and these are actively being used in India and other places. Apparently, this seems to be the only drug which is widely in use. Then there's another combination of drug, that is lopinavir and ritonavir. This is commercially available as Kaletra. This was initially, or actually it is used for treatment of HIV/AIDS. In SARS patient in 2004, it was suggested that it had some benefits. Based on that, it was also tested against the MERS coronavirus. In the animal model, it showed some activity. But when this drug was tested in a randomized clinical trial, and the results got published only on 5th of October in Lancet, it found the drug had really no effect. It did not reduce the mortality, it did not reduce the hospital stay or risk of progressing to invasive mechanical ventilation. These findings, therefore, do not support the use of this drug for treatment of patients admitted to hospital with coronavirus. Favipiravir is another drug. This is a nucleoside analog which has been approved for use in Japan to treat influenza. This drug has shown antiviral activity against West Nile virus, yellow fever virus, foot and disease virus. These are all RNA viruses. Based on these information, the drug was used in Italy extensively against coronavirus 19. A recent randomized control trial that has been done in India on 150 patients showed some benefit. It showed 28.6% patients showed faster viral clearance and 40% faster achievement of clinical cure. Hydroxychloroquine is politically charged molecule that was initially considered to be the solution for coronavirus 2. The drug has been in use for malaria and rheumatic disease. Initially, in the cell culture model, it inhibits virus replication very extensively. US FDA issued emergency use authorization, but subsequent several studies have shown that hydroxychloroquine does not significantly affect the hospital mortality or shows no benefit in COVID-19 patients. And in light of this, FDA has revoked the authorization of chloroquine, hydroxychloroquine as the COVID-19 treatments. This is a new molecule that has just been introduced. This is Ivermectin. It's a potent drug to treat parasitic worms. It has been in use in humans for river blindness. And veterinarians use this drug as a cure against heartworm in dogs. In the cell culture model, Australian researchers showed that this drug inhibited the virus replication very significantly. Soon enough, several clinical trials started. In Singapore, for example, this trial is running with 5,000 patients to see if this can prevent people from getting infected. So these are not patients. It's in 5,000 people, healthy people, to see if they can be, if the disease can be prevented in these people. A recent placebo-controlled trial, which is not yet published, but it has become available on websites in Dhaka. This drug has been used along with doxycycline for 5 days, combined with the standard of care treatment. The study demonstrated a clear benefit to treated patients. This work is not yet peer-reviewed and published, but it has given a lot of hope of utility of this drug. Nevertheless, the drug has been used increasingly in Latin America. Covalent plasma has been used extensively, is being used in India, but there is no, the data now, which is available also from India, clearly shows that there is no benefit to patients who receive plasma versus those who did not. Very recently, 2 companies have gone into clinical trials with monoclonal antibodies, Eli Lilly and Regeneron, and these monoclonal antibodies have shown some benefit in patients in initial trials. The large trials, which are placebo-controlled, are currently underway. So what are the efforts of DBT institutions in developing antivirals against SARS coronavirus 2? I have listed them over here. There are efforts being made to screen for inhibitors of virus replications using in vitro cell culture model. There are several efforts trying to repurpose the existing FDA-approved drugs. There is a lot of work going on in silico screening of SARS coronavirus 2 inhibitors. And of course, there is a lot of work going on development of therapeutic antibodies. What we have done at RCB over the last 6 months is developing an assay for testing the antiviral activity of molecules against SARS coronavirus 2. We are testing the activity of these molecules on live coronavirus in BSL-3 environment. So far, we have worked with about 50 pharma companies across the country and testing about 400 drug substances. I think about 3 or 4 molecules have shown activity which is very similar to remdesivir, and a couple of these companies are now planning to take these molecules further for clinical development. We are also working with the Ayush as part of a DBT collaboration with Ayush to test the herbal extracts and several other Ayurvedic preparations for antiviral activity. And there are some initial positive indications for some of the known herbs and the ones which are consumed in large amounts across the country. We are currently trying to see if we can identify the active principle which is inhibiting the virus replication, and we are hoping that in about 3 months' time we should have some answer to this. I think I will stop over here, and we'll take questions towards the end of the webinar. Thank you very much.
Thank you, Professor Vrati. That was a very comprehensive presentation on the drugs in line to treat COVID-19 and the efforts at RCB. Thank you very much. And I would now like to invite Dr. Subeer Majumdar to talk to us about the efforts at THSTI and NIAB. Over to you, Dr. Majumdar.
Yeah, I think Dr. Vrati has to quit sharing.
Yeah, I'm doing that.
Yes, we are doing that. So while Dr. Majumdar is putting up his slides, I would like to request the audience to start sharing their questions with us in the question box that you can find in your console.
Good morning, everyone. This is Dr. Subeer Majumdar. I will be speaking on behalf of National Institute of Animal Biotechnology, Hyderabad, and also THSTI at Faridabad. And this is about the response of DBT's AIs on COVID-19 part 3. And it's all under the governance of Department of Biotechnology. And it's my privilege to speak about both the institutions. So first and foremost, although we are Animal Science Institute at NIAB, we have done our share of responsibility towards COVID-19, and we all should remember that most of the diseases, specifically around 75% of the infectious diseases, come from animals to human, and that is why animals are equally important. So this institute has done its job. We have developed for detection a novel electrochemical device which is very ultra sensitive, and it is based on the graphene electrodes where a minor change in electric current can be noticed. And there what we have done is, you can see this is the COVID-19 antibody. It is stacked on the graphene electrode, and as soon as the antigen, which you can see in green color, goes and binds from the nasal swab, you will find a change in the electricity which is measurable. So this change in voltage, even small change, is measurable. So important thing is, usually such devices are able to detect somewhere in the range of nanomoles, but this device can go up to femtomoles, and this work is now transferred to a company called as Biotechnology Private Limited. In addition to that, what we have done is another technique for detecting the viral RNA. So we will take out the virus using magnetic beads tagged with antibodies on the surface, to the surface of virus. Virus will be removed from the sputum or nasal swab, and it can be either treated or it can be heated, and the RNA will come out. And these are called as small aptamers. They are kind of DNA, and they are complementary to this RNA. So then they will bind. As soon as they bind, and kind of a structure is formed here, and as usual, you can add cyber green, and they will start giving you color, which is measurable with a small instrument. So you can see very well that how you can see that the changes are occurring, and it is increasing with the rise in the number of such places where this aptamer has bound. Now we are going further on this, and we are trying to make around 11 aptamers throughout the RNA, so that we can see the color very nicely and very prominently. And this whole work can be done 2 to 3 hours of time, and it will be extraordinarily cheap. That is our motive in developing this. Now I will talk about the COVID-19 response from THSTI, and they have worked on several issues. One is diagnostic, which is aptamer based or visual PCR or ELISA based. Other is testing, which is there is a biocell lab who does lot of testing. I'll tell what that. There is an iLab, which actually is a mobile van and goes to villages in Haryana and checks the presence of this disease in society. Services are provided not only in the validation of the kit, but a lot of virus neutralization assays are done to find out how the active vaccines are developed and how those antibodies formed are good or bad, because you cannot work or experiment with humans. So those antibodies are brought to our lab, and there we check the interaction of virus and viral growth, whether that is inhibited by the antibody generated by a particular vaccine. Almost most of the vaccine people in India who are developing are doing this assay at our THSTI. We have also biorepository of the serum panels. There are a lot of medical jobs done at THSTI, and this clinical Dr. Sinjani Bhatnagar runs this, and very successfully. Earlier Dr. Kang was also there, and we have a lot of serum panels which are really helpful for several work. So this is that mobile van which has already covered around 5,000 RT-PCR results in villages, and in addition to that, there is also training program given to various hospitals in Faridabad, Gurgaon and Palwal by THSTI to conduct their own PCR detection, and Dr. Guru Prasad is the one who is doing it there. Now, when we go to other things, we have developed a highly specific and sensitive antigen test to the nasal swab, and this is again using a different kind of system where antigen is placed on the well of the plate, and then aptamers bind, and with streptadin, avidin, HRP conjugate, you can see the color reaction, and this can occur in 4 hours. There is a ELISA test for detecting antibodies generated, and this is also RBD based ELISA test. This takes around 4 hours. This is also licensed to a company called Exciton. And molecular diagnostic, conventional PCR based or visual detection is a very interesting thing, that you can really do this job and you can visually see the difference in the color for detection of the presence of SARS-CoV-2 RNA. This is also licensed to a lab, Genie Lab. And these are the people really who are very, very important for doing this, Tarun, Gaurav, Tripti and Guru Prasad. Now we come to support labs, and where a lot of vaccines which are developed, I said the antibody comes to us, and these are the normal cells, and when they are infected with the virus, they should ideally show a lot of pathological changes. But if you have added right kind of antibody, then you will see the cells do not show pathological changes, proving that the antibody generated either in an individual because of infection or in an individual because of the vaccine, that antibody is very good and supporting in fighting out the virus. Similar kind of test can be done to see the plaque assay. That means how many colonies of virus can grow without or with the good antibody, and that is called as the plaque assay for the reduction neutralization assay. And this is very well done in THSTI, and these are the people who work, and we are helping several companies who are developing vaccine to test the kind of antibody they generate in a human. In addition to that, there is a support system for product development, THSTI Bioassay Lab, IDRF, and biorepositories there. So we really help vaccine developers. We help in diagnostic kit development. We give control panels, that is serum, frozen swabs of the patients from nasal and oral, and we check the virucidal activity, antibody therapy, whoever is working. That means passive immunization. Direct antibody can be given when a person is infected to really calm down the activities of the virus, and that also we help at THSTI. THSTI Bioassay Laboratory is now working in the partnership with CEPI. CEPI is Coalition for Epidemic Preparedness Innovations, and this is really a very good thing which Dr. Renu Sarup, our secretary, has taken up. And CEPI has really helped us to make us one of the 7 labs in the world. With the same reagents and SOPs, we can do the work for testing of the vaccines to address vaccine development and manufacture. The beauty is then vaccine can be developed in any one of these countries, and that can be used in any other countries, because while developing vaccine, reagents and the protocols were standard used under CEPI. This is a very good approach which India has taken, and it's hats off to DBT for this. THSTI Bioassay Laboratory under CEPI partnership will provide ELISA of the spike and N protein, virus neutralization assays, pseudovirus neutralization assays, and ELISpot, basically Th1, Th2 response, and specifically measuring interferon gamma to show the T cell specificity of the viral action. There's a beautiful thing developed in THSTI. We call it hamster infection model. So what is hamster is just a model like human beings. And let us say that before infection or exposure of hamster to the virus, on day -1, we collect blood samples, and then we actually give the exposure to the virus. And these are the day 2, 4, 7, and 14. The spleen and blood is collected here. Lung and intestine are collected by day 7, and endpoint is on day 14. And what we check here, that if hamster is infected with virus, what happens? So that is shown in next slide. This is a healthy hamster, green color. You can see its body weight doesn't really reduce, and infected with 10 to the power 4 PFU of virus or 10 to the power 5 PFU of virus, you see there is a reduced body weight. In addition to that, if we check the gross pathology of the lungs, you will find untreated animals, the lung is healthy, but those which are treated on day 2, you can see certain changes. On day 4 and on day 7, you can see maximum changes in the lung, but with the grace of God, it comes back and it is okay by day 14. If you see the spleen also, these are the untreated animals' spleen, and these are the 10 to the power 4 and 5 PFU. You can see there is an enlargement of the spleen. By day 4, it increases, and by day 7, it is really increases high. But on day 14, you can see this, all this comes very close to the normal, which means that by day 14, the viral load decreases, and the patient or the hamster here is started showing the signs of recovery. This will match very well with the 14 days quarantine period, which we say to do for the human beings. So hamster is a really good model for undertaking studies of COVID-19. Here you can add various antibodies and check whether you can prevent these pathologies or not. For that, there are some other experiments done, and this is only showing the antibody titers. And most importantly, I will show this one. This is given in colored. This is day 0, green is day 2, blue is day 4, and this is sky blue is day 7, and this is day 14. So you can see that on day 2, maximum cytokine storming is taking place. However, IL-4 is giving a peak in cytokine formation somewhere around day 14. So this is the cytokine theory also proves well in this model. So it's a wonderful model. Almost every person who is developing vaccine in India, they are coming for testing this, whether it is Covaxin or now it is Sputnik is also coming, or the Cadilla's vaccine. Everyone is coming to THSTI for such kind of assays. In addition to this, there are several other companies which are really taking these services from us, and these are the ongoing, and these are to be done, and it's written detail, and just to show that how many companies we are helping. In addition to this, we are also working with DBT Ayush Network Program. Dr. Madhu Dixit is in charge of that, and this is recently started, and several formulations in addition to the herbal extracts are being checked for the antiviral activities. And there what we check is immunomodulatory activity of the herbal extracts, cytotoxicity assessment, anti-inflammatory effect, immunomodulatory effect on innate and adaptive immune response. And we also see antiviral activity and evaluation of the COVID-2 replication in vitro, and also the animal models have just shown. So there's a big DBT Ayush program network is going on at THSTI. In addition to this, Dr. Shinjani Bhatnagar is taking a lot of efforts in really collecting samples from the patients in various hospitals, specifically LNJP, ESI Hospital, and GCH, Al-Faha, SGT Medical College, and Medanta Hospital, LHMC, and around from 4,000 participants, she has collected 65,000 samples. And the beauty of that is that there are certain samples which are really come before the pandemic, so they are the pure control, and these all sera panels are available. Whichever company wants to take them and do some work, they can do. And recently, WHO has taken interest to help us in developing a pediatric COVID study to identify risk factors of severe COVID-19 in the children, which is a very rare thing, which is not done all over the world. So from these bioresources, several people can take RT-PCR confirmed SARS-CoV-2 positive samples. They can take sera panels, and they can take our services of evaluation of the panels, or nasal or oral swabs with the virus, negative sera samples for pre-pandemic. That means where the virus never occurred, and we have some samples at that time, so no way that virus was there, and those samples are pure samples. If I want to collect negative sera samples now, you have doubt whether the man was exposed to virus or not. Now we go to a very important study in the clinical side, and you can see there are people in blue color. They are kind of asymptomatic, what we call, and these are the people whom we call as symptomatic. If you see the antibody production in them, you will find those who were asymptomatic, only 60% of those people developed antibodies, and those which were symptomatic, around 90% of them have antibodies. So you understand what we are talking is asymptomatic people. It is not necessary that all of them have developed antibodies. Some of them do not. Worldwide, it is coming out to be around 10%, but in our country, it is around 30%. So it's a very important information given here. I go to next slide, and I show that these are the list of industries we are helping at this point of time related to COVID-19 work, and what are the kind of help we are doing is written here. But the bioresearch at THSTI Biorepository has received and responded to 32 requests from within academia and outside. So 32 is too big. We have only quoted 12 here. In addition to this is very important to show that we are working with Bharat Biotech Covaxin, that is the drug vaccine they are developing. Here we are working with Zydus Catila, Minvax, this is from Bangalore, Biological Evans, Indian Immunologicals Limited, and now I think Serum Institute from Pune has also come up with the passive immunization, and we are also working for with World Health Organization for CP, and it's a wonderful thing THSTI is doing, and we are grateful to the DBT's initiatives for doing such a nice work. I thank you here. Bye.
Thank you, Dr. Majumdar. I think that was a very difficult task to pack the work of both the institutes in just 15 minutes. It's really great to see this interdisciplinary and collaborative effort, global effort, coming up as a collective response to the pandemic. And thank you very much for that presentation. And with that, I would like to invite our third speaker for today's webinar, Professor Dinakar Salunke, Director of ICGEB Delhi, to share the work of ICGEB with us. Over to you, Professor Salunke.
Good morning to everybody. And since I am the third speaker in the series, my task is little easier. I think you have all the background information that is required. So I will not spend time in trying to explain to you introductory issues, but get directly into what ICGEB, the International Center for Genetic Engineering and Biotechnology, has been doing in last 10 months towards addressing this problem. ICGEB is focusing both on trying to get a quick solution as well as trying to understand the virus. And both ways we have been fairly advanced in our studies, and what I'm going to do in the next slide, if I can get to the next slide, is to give a list of get you the list of things that are being addressed at ICGEB in specific terms. So the one most important work that we have advanced at a significant level is a phytopharmaceutical drug candidate, which was initially developed as a solution, therapeutic solution for dengue, is actually being attempted for the COVID-19, and this work actually my colleague Naveen Khanna in ICGEB is doing in collaboration with Sun Pharmaceutical and CSIR, and DBT initiated us into this with dengue as a focus about 10 years ago. We have actually very early started developing monoclonal antibodies against COVID-19 as a therapeutic option. I'm going to talk about that also. Then there is integrative approach for analyzing genome, and that has also provided a lot of understanding of how this virus behaves. In terms of diagnostics, we have two approaches. One, a artificial intelligence based classification a colleague of mine is doing, and hopefully that will reach into a non-invasive method of diagnosing COVID-19. And there is other conventional approach of antibody test as well as antigen test, and it is being done in collaboration with the company which has taken many diagnostics technologies from ICGEB in the past and which are in the market. So the antibody test is already ready and being used, and antigen test is under development. In addition to that, as a part of the DBT's efforts for outreach, we have been doing assays for Delhi region samples for testing, and almost about 2,500 tests are being done per month. And we have actually addressed a requirement of the industry in terms of if anybody has a prospective compound, we are actually providing them our facilities, because we have now established assay for testing the drugs, whether they work in vitro on the virus or not, and those are being also carried out. So to first talk about the phytopharmaceutical drug candidate, which has been named by Sun Pharmaceutical as AQCH, is the reason why we decided to test it for COVID-19, although it was being at an advanced stage as a therapy for dengue, and I can tell you that it works for dengue. One of the main reasons was that both are positive stranded viruses, but the most important thing that we realized is that the common endosomal pathway for post-entry intracellular processing in both the cases, although the initial interaction with the host is through different receptors during the cell entry. And in addition to that, immune activation to increase the pro-inflammatory cytokines also is common in the both cases. And these two reasons is why we thought we should try this drug, which was ready in the initial stages, to try and work towards this. So the AQCH, the anti-dengue drug, which also works incidentally for chikungunya, which meant it actually had a broad spectrum of antiviral activities. And as I mentioned, in vitro mode of action of AQCH against dengue, and as well as it interferes at the endosomal release level, which is apparently similar to that in case of COVID-19 virus. And with these issues in hand, we actually got AQCH tested at our ICGEB Trieste laboratory in vitro studies, and it showed remarkable results. So the human safety studies for AQCH were already completed, and we were ready to go for the phase 2 clinical trials, and those are being done in 10 centers across the country, covering about 200 patients, and the results for this are expected by the end of this month, and hopefully we will have a functional drug for COVID-19. We keep our fingers crossed. We have, as I mentioned, been trying to make a therapeutic antibody for COVID-19, and that is being done by my colleague Anmol Chandele in collaboration with ICMR laboratory as well as Emory University USA, with which we have a long-standing collaboration. ICMR made very quickly available to us the samples which are 6 to 8 week post recovery SARS patients, and which should have high levels of antibodies. And using those samples, my colleagues actually used the B cells, memory B cells from there, and developed the monoclonal antibodies. And after sequencing them, actually they were able to express a large panel of antibodies and got them screened at Emory laboratory, and we have a very large panel of antibodies that neutralize in vitro the COVID-19 virus. So subsequent functional studies are being carried out towards making sure that in every possible way these antibodies work as a therapy. As I mentioned, as a possible diagnostic way, my colleague in computational biology has developed artificial intelligence based methodologies to analyze the chest x-rays from whatever source of data available to him, and developed a substantially functional model. The data he has been able to publish of using a training set and try and detect. It will not only detect whether the images have infection due to COVID, but other diseases also. And so he has 4-5-fold discriminations possible. The other studies in ICGEB are addressing not only SARS-CoV-2 genome, but protein and structure. And I have listed here many actually results that have been arrived. Many of these studies have already been published in range of journals. So more than 40 miRNAs with antiviral activity specifically directed to ORF1ab and S protein. There is a peptide antiviral that has been shown to have a potential for binding to S protein. This is in silico, and the network activities of identifying the interacting partners both at the microRNA level as well as protein level has been done, and the in silico screening of many lead inhibitors is being done for a variety of target proteins in against COVID-19. So lot of it in this case is in silico work, but there are collaborations established within ICGEB to take these things forward in an experimental model system. And thank you for your attention.
Thank you, Professor Salunke. That was a very insightful presentation, and you took us through the work being done in ICGEB. That brings us actually to the last session of the webinar, which is the question and answer session. And we have exactly 10 minutes, and very happy that we could finish it on time, and we have 10 minutes for question and answer session. So we have a couple of questions being dropped in the question box, so I will get on with them. Since we just heard Dr. Salunke talk about AQCH, there's a question to him. Since AQCH is showing activity against both dengue and COVID-19, in the clinical trials, is it also being taken up? Is there a possibility that in the clinical trials it's being tested in patients having co-infection of both dengue and COVID-19?
The present trials are focused primarily for COVID-19 because of urgency of the situation. So it is not being tested for co-infection.
Thank you. The next one is for Dr. Majumdar. How do the test kits developed at NIAB and THSTI compare with the rapid antigen test kit available in the market in terms of specificity and sensitivity?
I think this is comparable, but only time will tell, because these are all given to companies now. So they will really conducting, not only enhancing the product, but they will also be conducting the tests on human. And in my view, it is specificity and sensitivity should not be a big issue, because most of the things are very biotechnological things, very clear cut, very sharp. And if it happens, it will happen nicely. At the end of the day, it will be the one which is very cost effective, which is affordable by almost everyone, and that's going to stand and stay in field, market, or society, in my view.
Thank you. The next question is actually for both Professor Vrati and Dr. Majumdar. Our audience wants to know that know about the success stories from the in vitro and the in vivo testing work going on at RCB and THSTI.
I think it's as far as THSTI work is concerned, I think it appears that some of the vaccine are really coming on very good, but we cannot disclose the results to anyone in the audience. But yeah, things are very promising, and there is no reason again that they should not be promising, because if the vaccine is prepared in a nice manner, antibodies sufficiently generated, they can neutralize the virus, as well as in hamster assay also they will be giving a very good result. These are the assays which are done to ensure that since we cannot work with human, that we are indirectly conveying this is going to work. But actual success will come only when in mass scale human beings are immunized and rate of infection drastically reduces.
Thank you. Professor Vrati, would you like to add to that?
Yeah, I'll talk about antivirals. As I said, that we have tested potential antiviral molecules for over 50 pharma companies in the country. And there are 3 to 4 molecules which look very similar in terms of their in vitro efficacy against the virus. We are using remdesivir as a comparator here, which is the best molecule available right now for treating the infection. As Dr. Majumdar said, because this work we do with the pharma companies, we are under obligation to not talk about this work unless these things materialize into some sort of commercial product. But it is heartening to see that we have some potentially useful molecules that could perhaps be taken to the next stage of development. Over.
Thank you. We understand that there is some sensitive information which you will not be able to share here. This is a question again for Professor Vrati. You spoke about the use of Ivermectin in the case of COVID-19. So our audience wants to know what's your personal opinion on Ivermectin being used as a prophylactic drug.
Okay, I'm not a clinical doctor, so my personal opinion is only real personal. I cannot make any prescription on this. What I can tell you is that I have seen the data, and it shows very significant inhibition of virus replication. I also only reviewed the data from Dhaka Medical College, where they have used Ivermectin along with the standard care, and they show a very significant improvement in patients. So I remain hopeful, but I cannot make any prescription.
Thank you. Yeah, we have a lot of questions pouring down in the question box, but we'll not be able to take many. This next one is for Dr. Majumdar. Is there any study being conducted in animals which can tell us that how the disease is being transferred from domestic or pet animals to human or vice versa?
Yeah, I think it's initiated. My institute at Hyderabad, the one not THSTI but NIAB here, has received a grant from DBT BioTechnology to really look into all sorts of the coronaviruses which are going to infect the animals and how they can shift to human beings. In addition to that, DBT, especially Dr. Renu Sarup, has taken interest, and we have started a first One Health program at National Institute of Animal Biotechnology, where we have roped in not only all over country veterinary institutions, Northeast, and also All India Institute of Medical Science, Delhi, and Chennai, and also the All India Medical Science Jodhpur, where we are going to rope in human as well as veterinary scientists together with the health personnel of animals and human in the district level, and really going to now work very hard to pinpoint how it goes from animal to human and how it can be tackled. It's a new initiative started.
Thank you. This is a general question for all the three speakers today. Given the rate of the virus mutations, would you think that one vaccine could work against all the mutants? What's your opinion on that?
So can I take this, Dinakar? Okay.
Yeah. Go ahead.
Sure, please go ahead.
Yeah. The mutations, it is the receptor binding domain of the virus which is primarily responsible for generating neutralizing antibodies. And expectedly, that has been pretty conserved so far. There are a large number of mutations, since this is an RNA virus, that is not unanticipated. But most of these mutations are in proteins that are unlikely to affect the neutralizing antibody response. Actually, there is a study now, this D614G mutation, which has been talked about quite a bit. There is a paper already available where pseudoviruses carrying one or the other mutation have been generated. And in the lab, they have tested the ability of these two viruses to be neutralized by the same sera. And they have shown that if at all, the newly emerged virus seems to be neutralized better with the neutralizing antibodies, but certainly it is as good as the previous virus. So these mutations that have occurred so far, they don't seem to make any difference to the vaccine that would become available, targeting the spike protein, which will eventually generate the neutralizing antibodies. Over.
Yeah. Also, Dr. Soumitra Das, who is the director of NIBMG at Kolkata DBT Institute, while presenting to Dr. Harshvardhan before 4 days, he did mention that they have really screened several viruses and they found mutations do occur, but with the grace of God, as of now, it is not in the receptor binding domain against which the antibodies are generated. And this is very important. Also, I think we must go ahead.
You're done.
Yes, sir, I am done. I just wanted to say that before we close, we must thank our students, fellows, project fellows, and scientists who have really worked hard to do this service to the country. They should be really congratulated.
Just one word of caution is there are now some cases appearing which are of reinfection, and that is worrisome, because if there is a one infection earlier, then there are already antibodies there, and in spite of that there is an infection coming. So the simplistic solution in terms of vaccine, one has to watch it initially before we can say anything in terms of mutations.
Yeah, also if you see the work what Dr. Shinjini Bhatnagar has done and shown that some of the people are PCR positive, they are mildly symptomatic, and sometimes you call it asymptomatic, and almost 35% of them didn't develop the antibody. So next time you have to follow whether are they those people who are getting reinfected because they never produce good antibodies in the first place.
Thank you, Dr. Majumdar and Professor Salunke for taking up this issue of the asymptomatic infections also, because we had a lot of questions about them as well. And I think we have tried to answer many of the questions today. Thank you very much for your time and thank you for joining us today. I would just want to make one last announcement before we end the webinar today. The audience will be redirected to a survey page as we end the session. So I request all of you to please provide your valuable feedback about this webinar, and you can follow DBT social media handle and India Alliance social media handle for more information about these webinars. With that, I thank all the panelists today for their time and for sharing their work with us, and the audience for their patience and for their engagement with us. Thank you very much, everyone, for joining us. Take good care of yourself. Stay safe till we meet you again for the last webinar of this series. Thank you.
Thank you. Thank you.
Bye.
Thanks to all.